polysates are nothing but the neutrophils or white blood cells present in the egg. And third type is mesolesical, where you can see the moderate amount of yolk. So here you can see moderate amount of yolk present here. And the last one is megalesical, where you can see quite large portion of yolk is seen, especially in birds, fishes, where you can see this type of yolk in those eggs. Then the second basis for the distribution, second basis for the classification is distribution of yolk. So that means whatever the yolk present in the egg, so based on its distribution throughout the egg, so it is divided into isolesical or homolesical, and telolesical and centrolesical. So isolesical or homolesical egg is nothing but homo means same type or uniform. Even iso also same. That means same amount of yolk is distributed throughout the egg. The yolk is equally distributed in the whole egg. Example, so those type of eggs which are like alesical, microlesical, oligolesical, okay. So all these types of eggs, they belong to isolesical or homolesical eggs. Then next is telolesical. So here, so usually the eggs contains two poles. One is called animal pole and other is called vegetal pole. So here, in this type of eggs, the yolk is present on only one pole, not the either of the poles. Okay, only in the one pole you can see this yolk, and the other pole will be usually free. That means if the yolk is present in the vegetal pole, then the animal pole is free. If animal pole it is present, then vegetal pole is free. So that kind of eggs are called as telolesical, and usually we see yolk only in the vegetal pole. Okay, only little or no yolk will be seen in the animal pole. And next is centrolesical pole, means the yolk is present in the center of the egg, that is in the cytoplasm, which is surrounded by a superficial layer. This we can see in insects. Okay, this is the second type of classification. So here this is the diagrammatic representation. So here you can see we don't see any like differentiation in the yolk distribution. So it is distributed equally throughout the egg. Whereas if you see in the telolesical, you can see the first half is almost free. In the second half, you can see the protein yolk. At the top, you can see fatty yolk. And in the centrolesical, usually we see more black dots at the centers. Okay, so that means the yolk is more distributed at the center of the egg. Then, on the basis of shell, the another type of classification is based on the shell. So we all know egg contains shell. You can see the hen, like chicken egg. We see shell, but there is a classification, that is clidoic eggs and non-clidoic eggs. So in the clidoic eggs, we usually see covering or shell, hard covering. Okay, and these are usually seen in the eggs of the terrestrial animals. So the animals which are usually residing on the land or terrestrial animals, they contain a shell. So example, reptiles, birds. Okay. Then non-clidoic eggs are the eggs which is not having any hard covering, like all viviparous animals. If you see in human being, will the human egg contains shell? No. Okay, so that means in some animals there is no shell. Based on the presence or absence of the shell, there are two types, clidoic eggs and non-clidoic eggs. So this is a clidoic egg, chicken egg. So we have already seen in the last class, where we see different fetal membranes which are located inside the shell, like amnion, chorion, allantois, then yolk sac, all this we have already discussed. Next is on the basis of development of egg. So there are two types, determinate or mosaic type and indeterminate. So in certain eggs, every portion is predetermined with respect to its potential for further development. If any portion of such egg is removed, then the defective embryo will be formed. So that means, so if you see in an egg, each portion is determined. So what is determination is a prefixed idea, okay? So if we are determined to do some work, we will, so if we have strong determination, we will do that work, okay? So that is determination. So it is a strong will. So in the same way, in the embryo, each portion will have a particular fate, okay? So particular fate means they are supposed to develop into some organ or some tissue or some cell. So in such type of eggs, if suppose that particular portion is removed from the egg, then that particular portion is completely absent from the organism, which is completely developed. So I hope you understood the concept. That means if in an egg, the top portion is determined to become head and the bottom portion is determined to become a leg or hand or something like that. Suppose if the top portion is removed from the developing embryo, then in the organism or the baby which is formed, in that we don't see the egg, head, okay, because we have removed the portion of the egg or embryo which is determined to form head. So therefore that particular organ or tissue will be absent or completely missing from the developed organism. Okay. So this is because of the removal of certain portion reports in a permanent loss of organs from the egg. The remaining portion does not have the potentiality to originate the lost part. So we say that the egg which has the future determined potentially for development in the form of mosaic. So therefore this type of egg is called as determinate type of egg. So that means in simple terms, every portion of the egg has a fixed destiny or a fixed target. So each portion has to become certain organ. So if we remove any of the portion, that organ will be removed from the completely developed organism. Okay, that type of eggs are called as determinate or mosaic type of eggs. So this type of eggs are seen in annelids and mollusks. Okay. Second is indeterminate type. So in this, it is quite opposite to the determinate type, where we don't see any determination. Okay. So that means any portion of the egg can become any organ, but that depends on the time of the development. Okay. So in this indeterminate type, suppose if we remove some portion of the egg, the other portion will come and they replace the removed portion. Okay, that means in the final organism, which is completely developed, you will see all organs and all tissues intact. You don't see any missed organ or missed tissue. Okay, because the other portions of the egg can fulfill or can replace the missed portion. Okay, this type of eggs are called as indeterminate type. This we can see in amphibians. So this is indeterminate cleavage and determinate cleavage. In indeterminate cleavage, first cell division where one cell becomes two. And if these cells are separated from each other, each has the potential to develop into the entire organism. So this is indeterminate. So that means every cell has the potential to become the complete organism, and every this what is called the development or differentiation is based on certain chemicals which are generated inside the embryo. Okay, if we discuss more detail into developmental biology, we see some chemicals will be produced during development, and the gradient of concentration of these chemicals will determine which portion has to become what. Okay. Then determinate cleavage means after cell division, each cell is decided to become what? So one cell it decides to become brain, and other cell becomes liver, and other cell becomes lung. So if you remove the portion from where the brain cells are coming, so if you remove that, the brain will be completely absent because other cells are they have determined to become the other types of cells. So this is regarding the classification of eggs. Okay, so eggs are classified based on all these factors. So now we will discuss about developmental biology, and we also discuss these points there also. Okay, so now we will discuss about animal development and developmental biology. So first of all, the developmental biology is a science. It's a branch of science where we usually discuss about how an organism is developed from a single cell. So the origin of this particular branch of science itself is based on certain questions. So first is how different types or how a particular organism is developed from a single cell, that is zygote. What kind of mechanism is involved? So that is first question. Second question is how a single cell can give rise to these many different types of cells. We are getting liver cells, lung cells, heart cells, skin cells, blood cells. So how we are getting different types of cells? Okay, that is second question. Okay, so once we get different types of cells, they are not just random. So they are properly arranged in the organism. So how this arrangement is taking place? So what are the instructions which are arranging all these things? That is third question. Okay. So like we have some questions based on which the entire science has been developed, developmental biology. So to explain these questions, there were some theories which were already proposed. So during 18th century, we have one theory which is called pre-formation theory, where they have stated that in the embryo, we have a miniature infant. That means, suppose if we take a human being, so within each embryo, there is a small human being. Okay. Only thing is, whenever the embryo develops, this human being will become big and becomes a baby. That's it. So the entire structure, everything is already there in the embryo. So that is what the pre-formation theory explains. Like they have also given the example of Russian nesting doll. So what is this Russian nesting doll? Here you can see a small doll which fits into the somewhat bigger doll, but fits in the other bigger doll. So like that, so come after you see 1, 2, 3, 4, 5, 6, 7. So 7 dolls will finally fix into this 8th big doll. Okay. So that means the structure is same. The big doll and the small one both have the same structure. But only thing is the size is different. Okay. So this is big and that is small. So the same thing based on this idea, they have proposed that the big organism or the developed organism is present in the embryo in a small size. Only thing is the small organism is becoming big before birth. So that is what they have explained. And in another way, another scientist what they have proposed is the same small human being is present in a sperm. Okay. So from sperm, So it has been transferred to the embryo and it grows. But both of them were wrong. And another theory was proposed by Aristotle 2000 years earlier, and that theory is epigenesis. Here, what he explained is the form of an animal emerges gradually from a relatively formless egg. So, this is somewhat relative because egg doesn't contain any form. The egg doesn't contain any small human being or small elephant or so. We don't find any organism in the egg or embryo or sperm, right? It is formless. From formless egg, we get a animal, okay, a form. So microscopy allowed scientists to witness the progressive development of the embryo, thereby validating Aristotle's theory. So then they don't know actually like how it is developing. But now if we see like IVF treatments, all these have come, and every stage of the development we can see in under the microscope. So from a single cell, like from egg, then sperm, to fertilization, to zygote formation, then embryo development, every stage we can see. So after studying all these stages, they could confirm that, okay, Aristotle theory is somewhat related to the fact. Okay. So what are the different stages of development are? First one is fertilization. So fertilization in vertebrates is the union of two haploid gametes to reconstitute a diploid cell. Okay. So this diploid cell can become into a new individual. So here what happens? So usually in our body we have somatic cells and germ cells. So somatic cells are nothing but we have the cells which are not involved in reproduction, and they are usually having two sets of genetic information, two sets of chromosomes. They are called diploid cells. But the germ cells, in females, we see eggs, in males, sperms. So they are having half the set of the chromosomes when compared to somatic cells. That means only one copy of chromosomes are present in germ cells. Now these germ cells, eggs and sperm, upon fertilization, they will fuse and we will get a zygote, which is a single cell but having two copies of chromosomes, one derived from the father and other derived from the mother. And if we see, it looks very simple like fertilization. Okay, the union of egg and sperm. Fine, we are getting a diploid or a diploid zygote. But that is not that simple. So it is not a single step either. Okay, we have a series of steps which are involved in the fertilization. So what are these steps involved in the fertilization? We will discuss. So first is contact and recognition between sperm and egg. So we all know there are millions of sperms. Okay, so not all sperms fertilize the egg, but egg is only one, right? So not all sperms fertilize the egg. So there are certain mechanisms which will prevent the fertilization of egg by multiple sperms. So only one sperm can fertilize the egg. So how it is regulated? That is one thing. And once the sperm fertilizes the egg, okay, there will be a fusion of genetic material and the egg also becomes activated and the metabolism starts. So these are the events that take place. So first and initial step is acrosomal reaction. So the egg contains a membrane which is called as Zona pellucida. Okay, and the sperm which tries to penetrate the egg or which tries to fertilize the egg, first that has to meet this Zona pellucida. Okay, and then it has to enter through Zona pellucida. So how this sperm will become successful in fertilization? So here comes an organelle which is called acrosome. So we all know, like in normal cells, there are some organelles called lysosomes. So lysosomes will have some enzymes which will digest the other parts of the cell, which are like there will be a normal process called turning over. Okay, in the turnover, what happens? Like we will have the waste material in the cell. So after every after some period of time, all this waste material has to be cleared from cell. So that clearance is done by the lysosomes, which are like a pockets, which are having some enzymes. They will digest this some debris and waste material. So during evolution, these lysosomes have modified to become the acrosomes. Acrosomes will be present on the head of the sperm. So usually in school we will see the structure of the sperm, like it is there will be a head and there is a tail. So in the head we see this acrosome. So acrosome contains some enzymes. They are digestive enzymes. Okay, so now the enzymes which are present in the acrosome, they will digest the zona pellucida, the outer membrane of the egg, and then they will try to penetrate into the egg. This is called as the acrosome reaction, which provides the sperm with an enzymatic drill to get through the zona pellucida. And as this acrosome reaction progresses, the sperm passes through the zona pellucida, and then they will go into the plasma membrane, and the complete acrosomal contents are lost. Now, completely the zona pellucida is digested, and the anterior head will enter into the egg. Okay, and the constant propulsive force from the sperm's tail in combination with the acrosomal enzymes allow the sperm to move into the egg through zona pellucida. So once the sperm penetrates the zona pellucida, it binds and fuses with the plasma membrane of the oocyte or egg. Now, the other other mechanism is this egg will become, it will be in the inactive state, okay, until fertilization. So the egg is in the quiescent state. Quiescent state means inactive state, which is already arrested in metaphase of the second meiotic division. Upon binding of the sperm, whenever this sperm digests the zona pellucida and comes and binds to the plasma membrane of the egg, then there will be some changes that take place in the egg. They are physical and metabolical changes. This will allow the cell to undergo activation. And this will raise the calcium levels inside the cell. And now, from the second meiotic division, it will start continuing the next phase. So from metaphase, it will go to the next phase. So this is called cortical reaction. Now, what is the role of zona pellucida? Means the critical importance of zona reaction is to block polyspermy in the mammals. That means to prevent the fertilization of egg by multiple sperms, the zona pellucida plays an important role. So what happens if many sperms fertilize an egg? The DNA content within the embryo or zygote will become more. So which is not accepted in the nature. Therefore, such type of zygotes, they won't develop into the organism, or they will usually get aborted. And in some rare cases, you see like a baby is born with two heads or tail or something like that. Means there will be some malformations in the body, and even that those organisms also, they won't survive for longer periods of time. So this is all because of failure in this mechanism. So this zona pellucida, first of all, it becomes hard. So it is not that easy to penetrate the sperm easily into that. And the sperm receptors are present in the zona pellucida and they will get destroyed. Once one sperm enters the egg, so the receptors, everything will be destroyed, and therefore no other sperm can come and fertilize the egg. And once the egg becomes activated, there are 4 stages which are involved in the development. One is cleavage, patterning, differentiation, and growth. So what is cleavage? So now this egg will get activated, it starts dividing. So here mitosis and cytokinesis of the zygote takes place. So mitosis, where first the DNA content will become double, and cytokinesis means is nothing but the division of the cell. So one cell becomes 2, 2 becomes 4, 4 becomes 8, like that. So first it becomes a large cell, and inside that we will see number of small cells. And the genes of the zygote are not expressed at first. Only first division and cleavage takes place, but the genes are not expressed. That means no protein or nothing will be produced. And once the cleavage ends, you will see a ball of cells. Okay, it is all the cells will form a round ball. That ball is called as, that stage is called as blastula. Once a blastula or a ball of cells are formed, now they have to rearrange. So we need a head, hands, legs, like that. We need a shape, right? Every animal, if it has to develop into an organism, it will have a shape. So to get a particular shape, first of all, the cells has to arrange into some shape. So that is called as gastrulation, where it will form layers, 3 layers. Okay, they are called germ layers. So once the layers are formed, now we can see a direction or angles. They are called axes. So front and rear. So that is upper portion and the lower portion. They will become anterior posterior axis, and front and back portion, they will become dorsal ventral axis, and left and right sides. That means you see any organism, suppose human beings will have a top, will have a bottom, they will have right side, right hand, left hand. Okay, front will have like stomach, back. So like means we'll have some directions. Okay, so that direction, first it has to set the direction. Then it will try to form. Okay, at the top we need head. So like that it will has to divide. On the right side, one hand and one leg. Left side, one hand and one leg. Like that. First it will form the axis. Then it will start forming the organs. That is gastrulation. So once the axes are formed, they rearrange themselves into 3 layers. They are called germ layers: ectoderm, endoderm, mesoderm. So the outer portion of the layer is called ectoderm, middle portion is mesoderm, and inner portion is endoderm. So here the genes will get expressed. So here if you see late gastrulation in the frog. So in the frog, after gastrulation is completed, the outer portion is ectoderm, middle portion is mesoderm, and inside you can see endoderm. And this last portion is the future anus. So the anus will be formed in the future from that portion. And the middle portion you can see archenteron, which will form the gut in the future. So like that, first it will form a rough sketch of how the shape of the organism has to be there. Next is differentiation. So differentiation is nothing but till here, till gastrulation, there is no expression of genes. Okay, so that means every cell has a potential to become a complete organism. But after gastrulation, after the germ layers are formed, the genes starts expressing. When the genes start expressing, there will be a differentiation. Differentiation is nothing but it is like specialization. They are only specialized to do some function. They will have some specific structure and they will have specific function. So that type of change is called as differentiation. So the cells of the embryo differentiate to form the specialized structure and functions that are seen in the adult. That is called differentiation. Like now every cell cannot do everything, right? We have different types of cells. Neurons, blood cells, skin cells, muscle cells. Now if you take a blood cell and you give in the brain, it cannot function like a neuron, right? So neuron, you have a structure. It has a function. Blood cell, you have a structure. It has a function. Okay, muscle cell, it has a structure. It has a function. So like that, every cell has a structure and a function. So that type of cells are called differentiated cells, and the process of formation of these cells are called differentiation. And these cells will organize into tissues. Then these tissues will together form an organ, and organs together form organ systems. So that formation of organs and organ systems is called organogenesis. And once the organ systems are formed, all together they will function into a complete organism. Okay. Then once the structure of an organism is formed and every cell type is differentiated, tissues are formed, organs, organ systems, everything is ready. The next stage is growth. Okay. So if we see in the human beings, till 3rd month of gestation, complete organism, you can see the structure, how the baby looks. But that doesn't mean that the baby is developed. Okay. Then the stage comes that is called growth. Okay. Till 10th month, okay, all these 10 months, there will be growth inside the womb of the mother. So this is by forming various extracellular matrix, all these new cells. Okay? Some old cells will be removed by apoptosis. So there are various mechanisms which function together to form a complete organism. So this is for the idea of axis. Okay? Anterior, posterior, then dorsal, ventral. So these are the axes which we have discussed already. Now, these germ layers. So there are 3 germ layers which we discussed just now. Ectoderm, endoderm and mesoderm. They will completely form the okay. So from ectoderm we get sorry. Yeah. So from ectoderm we get skin, brain, spinal cord, all other types of neurons, sense receptors for taste, smell, all this we get from the ectoderm. And from the mesoderm we get notochord, muscles, blood, bone, sex organs, all this comes from mesoderm. And from endoderm we usually get the lining, inside lining of the gut, the lining of the lungs, lining of bladder, liver and pancreas. These are all originated from these 3 germ layers. Now, we will discuss a bit about eggs and zygotes. So already we have discussed like various types of eggs are present, classified based on the yolk, distribution, yolk amount, all these things. So it's the same thing, like the size of the egg also it varies. Not only the yolk, the size of the egg also varies from organism to the organism. In sea urchins, if you see, They will be approximately 70 to 150 microns. And in humans, they are 100 microns. In frogs and fishes, then insects, we can see a bit larger eggs, were 1000 to 2000 microns in size. Then in birds and reptiles, they are millions of microns. That means in centimeters. You see big egg. So these eggs, they will have the yolk. So this yolk will provide the nourishment. So yolk is made up of lipids, carbohydrates, and protein, which are organized in the form of granules. So they will nourish the organism, okay, until the birth. So we have two poles. One is animal pole and vegetal pole. So this yolk, it settles at the bottom of the egg, and this bottom portion of the egg where we have complete yolk, that is called as vegetal pole, and the upper portion of the egg where there is no yolk, that is called as animal pole. Okay, so based on this axis only, that is the upper portion is animal pole and bottom portion is vegetal pole. So based on this axis only, we get all these directions like ventral, dorsal, posterior, anterior, all this will be designed. So eggs have different amounts of yolk. So the larger animals there, which are growing outside the body of the mother, okay, like egg-laying animals. So they have to grow outside. Okay, there is no nourishment from the mother. So in such cases, we see a large amount of yolks. But in animals which are growing inside the womb of the mother, they doesn't require yolk. Okay, so they will directly get the nourishment from the mother through placenta. Okay, and some animals, they usually grow as a larva. Okay, and frog and fish, they will have intermediate egg size and yolk content. So that means different animals based on the requirement, they will have the size of the egg and yolk content. And almost all of the zygote volume comes from the egg. And it will give the animal and vegetal poles of the egg. Okay. So here is the structure. So the top portion is animal pole and the bottom portion is vegetal pole. And inside we'll see the gradient. Okay, it is there will be color gradient, and that center portion will be gray in color, that is called gray crescent. Now we see the embryonic development. So from a single cell zygote, a complete embryo is formed. So that embryo is formed from the continuous division of the zygote. There is a rapid cell division, which is called cleavage, and that cells will form into a ball of cells, blastula, and finally will form an embryo. So how this cell division takes place? You can see here a single cell. At the center, we can see a contractile ring, which will be formed, and that will constrict the bottom, and that will divide the single cell into two cells, and later the two cells will be cleaved by a cleavage furrow. Okay, cleavage furrow is formed. It's a structure just between the cleavage, just before the cleavage of the cell. Then, after division, they will be forming a blastula, a ball of cells. Okay, so how it looks like is the cells are rearranged themselves into a ball, which is called blastocyst or blastula. Okay, in human beings we see blastocyst. Okay, in frogs also we call them blastocyst. So that they will contain a fluid-filled interior, which is called blastocele, which will be surrounding by a layer. So this is how it looks. So the red color cells you can see, they are called blastoderm cells, which forms mesenchyme, and the portion below this layer is called as blastopore. Now these blastoderm cells, they invaginate, they will form a fold and they will extend to form archenteron. So archenteron, which we already discussed, like they will form the gut in the future organism. After the blastula is finish the wall folds inward at one point, and it will form a tube-like structure which is called archenteron or primitive gut. And the opening of archenteron is called as blastopore. And the cells at the animal pole, they will grow and spread over the outer surface, forcing other cells to invert. So it is nothing but it will form like a tube-like structure into the cell, into the wall of the cell. Okay? So this you can see, the cells at the sides of the archenteron, they will elongate and they will form a tube-like structure. You can see the extension, and you have a blastopore at the bottom. Then you have the primary mesenchyma at the bottom, the red cells, and the top one is secondary mesenchyma. Here you can see the ectoderm, which is outside layer, and the endoderm, which is the innermost layer of cells. Okay, this is how division, fertilization, then division, then rearrangement. Okay, all this takes place. Now you got the germ layers also. Now from these germ layers, there will be differentiation and organ formation, which is called organogenesis. So this organogenesis we will discuss in the next class. Okay. Any doubts? Okay, if you don't have any doubts, we will continue this topic in the next class. Thank you.
हेलो मैम,
ये क्या है, बात करके
मैम
चले
गए
भाग गए मैम।
चले गए
हाँ
चले
तो
गए
यार
चले
वो अब इम्पोर्टेंट क्वेश्चंस कौन देते हैं? अब
टैग
ये
करो ग्रुप
VRA
में
के
दे
होस्ट
दे।
देते क्या?
कल पूछ लो।
नहीं, ग्रुप में भी बोलो, उनको टैग करके बोलना पड़ता है।
ग्रुप में नहीं बात करते कौन से भी सरा मैडम,
वो तो खाली
ग्रुप
रिप्लाई
में
देते हैं।
बात करना शर्माते जी।
कल पूछ लो, क्लास स्टार्ट होते ही पूछो।
सुनते भी नहीं, खामोश, पहले क्लास करो बोलते हैं।
उनको बोलना, उनको आप चले जाएं, कैसा करना हो।
बुलाने का
ऐसा
तरीका
भाग
है तो
गए,
कौन सा
समझ नहीं आ रहा
बुलाना था।
गुस्सा में। व्हाट्सएप पे कॉल करके बोलना उनको, मैम फिर से ऑनलाइन हो बोल के।
पूरे एकदम कॉल
नहीं
करना।
इंस्टाग्राम पे करो।
उन्होंने
हाँ
आईडी
आईडी
नहीं
है
दिया ना।
इंस्टाग्राम,
है उनकी आईडी।
वो भी मालूम कर
अब
रही
आप
है
टॉपर
सब।
आए, टॉपर को सब मालूम होगा।
है मेरे पास ऐड है, धतरी माधुरी बल के फोटो भी है उनकी।
अरे अरे वर्क कर दे रहे तो मुझको यार।
अरे है तो है जी भाई।
मिल जाते लो सबको, जल्दी से मिलते हैं। लास्ट में हमारे मालूम होते क्वेश्चन।
कहीं को ग्रुप में नहीं है
तो भी
मंडे रजिस्ट्रेशन करने जाना है।
ठीक है भाई, कब तक आना है बोला?
सुबह 9 बजे वहां रहे तो बस से।
क्या करते 9 बजे जाके दिन के वास्ते
गार्डन है ना,
पूरा साफ
टहलेंगे
साफ कर चुके हैं यार। झाड़ू वाड़ू मारो जाके 9 बजे, कुछ भी कोई भी नहीं आते यार मंडे उतने जल्दी।
पोछा भी मार लेना।
अपन जाके थे तो सुबह आ जाते।
उफ
मैं
उफ।
तो फोन करके बुलाएंगे।
क्या टाइम पे रहना वहाँ पर?
1 2
टाइमिंग बोलो, टाइमिंग कब तक रहना यूनिवर्सिटी में?
कुछ भी खास टाइमिंग नहीं बोलो जी, जस्ट चले जाना
11
है
बजे
बोल के
चल
बोल
गई बस है।
10-11 बजे रह सकते हो आप।
सिर्फ रजिस्ट्रेशन कराते कि भगा देते।
आपको रुकना है क्या वहाँ पे?
अपनी मर्जी पे
नहीं यार, रुकना वगरना नहीं रहता लोग। उधर रजिस्ट्रेशन जाओ जाओ करता लाइफ देखो।
बस
ठीक है, कल मंडे के
जरा
दिन
दम करना
कल
शुरू
लास्ट
करना है
क्लास है।
लोग को अब। दियो
हाँ
दियो दियो क्वेश्चन बोल के।
कल लास्ट क्लास में पूछना, दम कर देना फुल। सुना तो जम्बुल भाई
कल
स्पैम कर
स्टार्ट होते
देंगे
ही
अपन।
पूछना।
बॉटनी
स्टार्ट
के साथ भेजते बोले क्वेश्चंस पर्सनली।
पर्सनली क्यों
मेरे
ग्रुप में
पास
भेजना?
मटेरियल
ग्रुप में
नहीं है।
बात नहीं, ग्रुप में पूछे बोले तो या पूछे तो कैसा
नहीं,
शर्म आती
कोई
है
एक
मेरे
जने
पास
भेज
ग्रुप
दो,
में।
उनको भेज के उनका दिमाग खा लो। भेजो भेजो भेजो, रिप्लाई दो दो बोल के।
नहीं सेंड करा उनको तो अच्छा ठीक है, मैं भेजता हूँ मिलके बोले। नंबर है मेरा उनके पास।
नहीं, थोड़ा सा लाइक थोड़ी देर देखो। एक दो दिन में नहीं भेजे फिर से बोलते जाओ बोलते जाओ। पैसा निकलता है।
केमिस्ट्री के सर कुछ भी ना बोले?
केमिस्ट्री के सर कल बोलते बोले इंपॉर्टेंट क्वेश्चन।
केमिस्ट्री के सर दिए ना इंपॉर्टेंट क्वेश्चंस।
कब दिए?
अच्छा अच्छा, ठीक है।
अगर किसी के पास भी इंपॉर्टेंट क्वेश्चंस आए तो सेंड करो, अगर मेरे पास भी आए तो मैं सेंड करता हूँ।
सीधा ग्रुप में डाले तो किस्सा खत्म।
हाँ ओके, इंशाल्लाह।
ठीक है, तजम्मुल भाई लेफ्ट करेंगे अपन ना भैया।
लीव कर रहा हूँ मैं तो।
सोच लिए चलो भैया, दोनों एक साथ लीव कर देंगे। 1 2 हाँ करेंगे।